Hi guys, welcome to lecture 4. Thank you so much for being flexible and trying out this record a lecture thing. We'll see how it goes. Firstly, sorry that it's a little bit dark in here, but hopefully everything else will be fine. So, hopefully you guys are like at home chilling right now. So, grab a coffee, let's get started. Today, we're first actually going to reiterate some of the main points from last week's lecture. And then we'll dive into today's focus, which is going to be energy and life. So firstly, your Mastering Astronomy homework 1 is due before lecture next week. So before April 3rd at 1 p.m. It's 25 questions, 30 points, and if it's turned in late, there will be a penalty. So here again is a schedule for free tutoring, which is available every day in 165 if you want to go. So last week we talked about the composition and origin of the universe, and we looked at this picture, which I mentioned, but I feel like I didn't go into too much detail. So we know that it's called the Hubble Ultra Deep Field image. It was taken by the Hubble Space Telescope between 2003 to 2004, and we know that nearly every single little tiny dot in this picture is an entire galaxy of stars. So we estimate that there's about 100 billion stars in the universe. It's a very rough estimate. And this image contains about 10,000 galaxies. But even though it seems like a lot of galaxies, it's actually just a tiny little piece of the space, and it's in the constellation Fornax. So to get an idea of just how small this region of space that this image covers, imagine holding a grain of sand at arm's length looking at the sky. This entire image can actually fit behind that grain of sand. So let's also talk about why we call this ultra deep field. And that's because this is actually the deepest image of the universe we've been able to take. And it's allowed us to see the most distant astronomical things ever detected. So specifically, right? The nearest of these galaxies are actually millions of light years away. For example, like this little spiral galaxy down here, which scientists have named UDF 423. And then the most distant ones, the tiniest little dots that it's barely conceivable, it's around 13 billion light years away. So that equates to, right, the distance that light can travel in 13 billion years. And so that means that we're actually observing the galaxies. When we're looking at this image, we are looking at potentially 13 billion years back into time. So we're looking at the time just shortly after the birth of the universe. I mean, to me, that's just mind-blowing. So we also, in last week's lecture, talked about the concept of humans as star stuff. We talked about the idea, right, that all basic ingredients of the universe is constantly being recycled. So we talked about, right, in the early universe, hydrogen and helium were the only elements that was around. They kept nuclear fusion inside cores of stars that gave stars energy and kept them alive and shining. It was through this fusion that heavier elements like lithium, lead, and neon were made. And then when the stars eventually ran out of chemical fuel, they would explode in a supernova. And then they would scatter the elements into space and eventually become part of new clouds of gas and dust, from which then new stars can be born. So this life cycle of a star continues from one stellar generation to the next. So by the time our solar system was finally made about 4.5 billion years ago, about 2% of the original hydrogen and helium from early universe had already been converted to heavier elements. So what this means is that our planet and our bodies are made from elements that were created billions of years ago. So we are all stuff of stars. We reside in the universe, and the universe made us. But the universe is also a part of us. So we can, kind of think of those 2 points that I talked about as, some of the most integral things to understand about the universe. So we'll keep that in mind, and then we'll get into today's lecture. So today we're going to be focusing on energy and specifically energy, we know, right? There's different types of energy, but mainly energy incorporates just the idea of, but essentially energy is what makes matter move, and it can take different forms. There's kinetic, potential, and radiative energy. So kinetic energy is your energy of motion, which is anything from cycling to breathing. And then we have potential energy, which is stored energy. It can later be converted into other types of energy. So for example, a ball on top of a table has gravitational potential energy because if it rolls off, it will fall. And so once it falls, the potential energy that it had will be transformed into kinetic energy once it falls on the ground. So our example here is gasoline. And gasoline is potential energy because it contains chemical potential energy in the forms of little hydrocarbon chains that can be converted into kinetic energy when the car starts moving. And then energy carried by light, like sunlight, is radiative energy. We use the word radiative as a synonym for light. So this type of energy moves at a speed of 3 times 10 to the 8 meters per second, and it's what we're going to be focusing on in the next few slides. All right, so light, as we know, is both a wave and a particle, and the particles are called photons. So its wave is made up of a combination of electric and magnetic fields, which is why we call light an electromagnetic wave. So the wave is characterized by two things, a wavelength and a frequency, which are both related by the speed of light. So the frequency, which you'll sometimes see as written as the Greek letter lambda, is the distance between adjacent peaks of the electromagnetic field. So obviously, we can measure between the two peaks for the wavelength, but we can also take the trough to measure the wavelength. And then frequency is measured in 1/second, which is hertz. So the number of time each second that electromagnetic field vibrates, it's the wave's frequency. And we'll visualize it later so that it's a little bit hard to visualize it right now. And the two properties that we talked about multiplied together equals the speed of light. So essentially, if you know the wavelength of light, you can figure out its frequency as well. So light can have any wavelength, frequency, or energy. And the electromagnetic spectrum encompasses the entire range of possibilities. But humans are mainly familiar with the visible spectrum here, since it's the only type of light that we can actually see. So since the wavelength of light is measured in meters, we see that the wavelength of visible light is a little shorter than 10 to the -6 meters, which is around 1,000 nanometers. And nano stands for 10 to the -9. So we just use nanometer just to make the terminology easier. So specifically, the visible spectrum ranges from 400, which is violet, to 700 nanometers, which is red. And the difference in wavelength of visible light will come in play later when we talk about observations of stars. So here's the complete electromagnetic spectrum in all its glory. We can immediately see that our visible spectrum is only a tiny section of the entire spectrum. So this is the only portion of the spectrum that we can see. And there's all of this that we cannot see, but it does impact our bodies and our lives. So we can immediately see, right, that these waves, they are very compressed here and they get elongated as we go to the right. And so what this means is that the wavelength grows as we get to the right. So let's start with our visible light that we can see. If we go to the left, we reach ultraviolet. So you guys are all familiar, ultraviolet, it's UV, right? So the sun emits UV light, and it's called ultraviolet because it's on the other end of our violet spectrum, right? So the sun is our cosmic source for ultraviolet light, And if we don't protect ourselves from this UV light, then we can get skin damage and potentially even mutations in our DNA. And then as we get more to this side, we can see that the wavelength is getting even shorter than it is here. But we know if the wavelength is getting shorter, the frequency is increasing. So X-ray has a high frequency and a short wavelength. And so that's how X-rays are able to penetrate our skin and actually look at the composition of our bones. So this is what we use in X-ray machines. In black holes, they also emit X-rays. And as we get to the very end, gamma rays are the highest frequency light that we've detected. And thus, it's also the lowest wavelength. So gamma rays are 10 to the negative 12 meters. And we've been able to detect that in gamma ray bursts in the universe. So as we get to the other side, infrared light is called infrared light because it's on the other end of the red visible light. And infrared light can be emitted from anything like our bodies. So infrared light is a source of heat. If you put infrared light on, a human body, you can see that we are emitting and radiating light. Infrared light can also be detected in planets. Planets, star-forming clouds, they all emit infrared light as well. And then microwaves, we're all familiar with microwaves that can heat our food, right? And so what microwaves does is it's able to create vibrations inside water molecules. And so these vibrations then, move and they can start to heat up. So that's how we are able to heat up our food. And it's interesting to look at the size of these waves. So these wavelengths, right, it ranges from even smaller than a little hydrogen atom to radio waves, which is as long as a football field. And so when we talked about SETI, the search for extraterrestrial intelligence, we said that they use radio waves to detect any sort of communication that extraterrestrials might be using to communicate with us. And so that's why we use radio waves, because they have the longest wavelength and they can reach the longest distance. All right, so server question 1, I'm going to give you guys the answers and the questions, and this is just to make sure that you guys have watched the lecture. So is light a particle or wave? And we know that it's both composed of a wave and photon, which is a particle. So it's composed of both. All right. And one way for us to utilize light is to learn through something called spectroscopy. Spectroscopy involves collecting light through a telescope and then dispersing it into a prism to study it. So for example, if we shine a hot object like a light bulb through a prism, we can see that it emits light in a continuous spectrum of rainbow, from purple all the way to red. In astronomy, this allows us to study a distant object's temperature. And so this is what we call a continuous spectrum. But if we wanted to study a cloud of gas in space, let's say, atoms in the gas cloud emit light only at specific colors depending on the cloud's composition and temperature. So we would see an emission line spectrum down here with a background of black and then with bright emission lines at specific wavelengths. So the opposite would be the absorption light spectrum down here, which shows a background of rainbow but with dark absorption lines at specific wavelengths, which occurs when light from a hot source passes through a gas cloud. And what happens is a certain of the lights is blocked or absorbed, which occurs when light from a hot source passes through a gas cloud. And what happens is some of the light is blocked or absorbed by the atoms in the cloud. So it doesn't show up in the spectrum. So that's why we see a continuous spectrum here with thin black lines. And so those thin black lines were light from the source that was blocked out by the particles inside the cloud. So we can use all of these to detect the temperature and chemical composition of distant objects in the sky. And so this method is actually what we use to learn the composition of stars, gas clouds, and planetary atmospheres. So here we've been able to plot a black body curve, which amongst many things, it can show the relationship between a star's color and its temperature. So we can see here, our star is a medium-sized and medium temperature star. So it's seen in the yellow spectrum. And this Hertzsprung-Russell diagram here is another way of showing the same relationship. The blue stars are right here, if I can find my browser, the hottest stars and one of the brightest. And we can see here, the red stars are the coolest and most dim. And then the luminosity, right, the brightness of the star increases by size as the giants are a bit more luminous and the supergiants are the most luminous. So scientists can infer even more information about distant objects. And perhaps the most important is the Doppler effect, which allows us to detect the slight motion of a star caused by an orbiting planet. So let's say we're right here on Earth and we're looking at the motion of a star far away. So as the star moves away from us, we observe what's called a redshift. And the redshift is when the wavelength of an object increases as the distance between us, the observer, and the star is getting bigger. So when it's moving towards us, however, we can see that the distance between us and the object is getting shorter. And so the wavelength detected is getting shorter. And so, for example, the Andromeda galaxy is detected to have a blue shift. And so we know that it's moving closer and closer towards our galaxy. So here you can see the spectrum of some of the most common elements and just how much they differ. So we know that even though they're composed of the same things, proton, neutron, electron, the way that their chemical fingerprints show up is very different between the different elements. All right, now that we've talked about energy, let's talk about the consumers of energy, living organisms. So let's discuss what life is, right? Here we have a cat and a cat car. So they both have a lot in common, right? They both require energy to function. The cat gets its energy from food, and the car gets energy from gasoline. They both can move at different speeds, and they both expel waste products. But we clearly know that the cat's alive, but the car isn't, even though, let's be honest, sometimes it seems like the car's doing more than the cat just laying there lifeless. But what separates them, right? What makes it that the cat is alive and the car isn't? So we know that life on Earth is really diverse, as seen here. It includes 3 domains: bacteria, archaea, and eukarya, and they all originate from one common ancestor. But what links all of these living things together? So here we have the 6 properties required for something to be considered living. So first, order. So just like how molecules are neatly compartmentalized in cells with specific areas for DNA production in here, energy production in the mitochondria, and degradation of waste products in the lysosome, we need order in order to have life. And like the definition given by the Institute of Astrobiology we spoke about earlier, reproduction is another key property. So living organisms can either reproduce or they can be products of reproduction. So some organisms like mules, they can't reproduce, but they are still products of reproduction because they're offsprings of horse and donkey. But they themselves cannot have babies. So then third, growth and development. Living things grow as directed in part by heredity, which are traits passed to an organism from its parent. So everything passes its heredity through DNA. Some things like viruses pass their information through RNA, but we don't actually consider viruses a living organism. And 4, energy utilization. So living things use energy to maintain order in cells to reproduce and to grow. Life without energy is not possible, and organisms can get energy through either food or chemical interactions with their environment. And the environment in turn gets its energy from an internal source, like heat of the planet itself, or an external source, like sunlight. Okay, 5. Responds to environment. Living things interact with their surroundings and they respond to environmental changes. So, for example, warm-blooded mammals like us, we sweat to maintain a constant body temperature. So that's how we respond to a hot environment. And then lastly, evolutionary adaptation, which is evolution. So life evolves as a result of interactions between organisms and their environments, and that leads over time to evolutionary adaptations that makes the species better adjusted to their specific environments. We've been referring a lot to the word species, but let's define it. So species is a group of organisms that share some set of common characteristics and are able to interbreed with each other in order to produce a fertile offspring. So in order to write the species names, scientists actually came up with a universal system using Latin base so that their scientific names don't get lost in translation. So when we write it, the first part of the name is its genus, which is the generic name to which the organisms belong. And then the second part is the specific species names. And it distinguishes which type of species is the one that we're talking about. And we always write the full name in italics, and we always capitalize the genus name. So for example, we belong to the genus Homo, and specifically we are the species sapiens. So humans are called Homo sapiens. We, in the past, have had a lot of other brother species and sister species, but they have all gone extinct. So we are now the only members of the genus Homo. Another example, horses and donkeys. Horses and donkeys, they both belong to the genus Equus, but their species names are different. Our modern understanding of evolutionary adaptation is described by the theory of evolution. And evolution just means change with time. But before we had this theory, the idea goes back about 2,500 years ago with Anaximander, who we talked about in lecture 2. And he also promoted the idea that life originally arose in water and then evolved from simpler to more complex creatures. So Aristotelians, which was led by Aristotle, they became entrenched within the theology of Christianity. And they maintained the idea that species are fixed, independent, and do not evolve. And so because they held so much of the power, evolution wasn't really taken seriously for another 2,000 years. And a while after that, a guy named Jean-Baptiste Lamarck, he came up with the idea of semi-evolution. He believed that traits are passed down. But in his mind, traits can be acquired. And he thought that, for example, if a parent developed muscles, their offsprings will also inherit those muscles. So we know that that's wrong, but Darwin said that traits inherited were innate traits, not ones developed. So here he'll go into detail about how he found that out. So in 1859, Charles Darwin published On the Origin of Species, which used observations of animals and fossil evidence to support the idea of evolution. Some say his model was built from 2 undeniable facts and 1 inescapable conclusion. So the fact 1 was that overproduction and competition for survival. What this means is that parents overproduce children in hopes that the children will be able to survive in this competitive environment. So it led to this question of why is the environment competitive and why don't all of the offspring survive? And fact 2 is individual variation, in that some individuals possess traits that make them better suited for the environment and better suited to compete for food and shelter in that area. So the conclusion that he was able to make is that there is unequal reproductive success. So in the struggle for survival, some individuals will survive and some will not. And it's dependent on how well adjusted their traits are to the environment that they live in. Okay? This unequal reproductive success we call natural selection. So this is where advantageous genetic traits will just naturally win out or be selected over less advantageous traits, just because they're more likely to be passed down through generations. Organisms with those advantageous traits are more likely to survive, right? So Darwin was able to find evidence for evolution by natural selection when he went to the Galapagos Islands, and he saw all the varying types of finches and how much their beaks were different. So he presumed that perhaps an ancestral pair of finches reached the Galapagos Islands from the mainland, perhaps. And over time, local populations of these island finches gradually became more adapted to the distinct islands that they were in. And so their beaks, some, if they were seed eaters, became larger in order to accommodate, being able to eat those large seeds. And then you see these little flower eaters. Their beaks were distinctly smaller and more precise, more sharp than the seed eater beaks, because the flower eater beaks were more adjusted to the environment that they were in. So let's look at an explanation of Darwin's finches.
What are Darwin's finches, and why are they so important to biologists? The study of finches led to the development of one of the most important scientific theories of all time. But how did this come about? In December 1831, a naturalist called Charles Darwin boarded the HMS Beagle bound on a surveying voyage to South America. Whilst the ship and crew carried out coastline surveys, Darwin was free to explore the islands en route. In 1835, the Beagle arrived at the Galapagos Islands near Ecuador. What Darwin found there surprised him greatly. As well as giant tortoises and marine iguanas, Darwin collected and preserved a variety of different songbirds called finches. Upon returning to the UK, he examined them together with ornithologist John Gould and made some fascinating discoveries. The scientists observed that the birds were all similar to a single type of finch found on mainland South America, suggesting that these mainland finches had originally colonized the island. However, the Galapagos finches were all slightly different from the original mainland species, and they were also different from each other. The finches on each island showed distinct variations in their overall size, beak shape, and claw size. These differences were attributed to the differing food sources available on the various islands of the Galapagos. Some of the birds had long, thin beaks and sharp claws suited to catching and eating insects, while others had large, powerful beaks suitable for cracking open nuts. Because of the distances between the islands, breeding between different species of finch was unlikely, and Darwin concluded that the finches must have evolved over time from the original mainland species to suit the conditions found on each individual island. In all, 13 of the birds brought back by Darwin were identified as being entirely new species, all similar to each other, but with definite variations from their common ancestor. Darwin proposed that the variations seen both within and between the finch species arose by chance. Variations which gave any individual a competitive advantage made them more likely to survive and therefore reproduce, outcompeting those with less advantageous characteristics. Darwin called this theory natural selection, and he published it in his book On the Origin of Species in 1859. Evolution by natural selection is now widely agreed to be the most accurate theory to explain the origin and diversity of all life on Earth. In this video, you have learned how the finches of the Galapagos Islands led to the development of the theory of evolution by natural selection. Is evolution of these birds still happening? See what you can find out.
So what Darwin was able to see in the finches, we've been able to see in many organisms in nature. Tree owls, for example, the traits they had were favored in certain environments over others. And so in a tree, they are much more likely to be able to camouflage and survive than something like a snow owl. And this idea of evolution applies to humans, obviously, as well. We are how we are now because our ancestors were better suited to their environment than some of the other creatures around. So we were able to populate more and more and eventually get to how we are now. Although some would say we are not exactly evolving to be better. But back to Darwin. Another way that he was able to find strong support for his theory of evolution was by looking at examples of artificial selection, actually. And so that's the selection of breeding domesticated plants or animals by humans. It's kind of like humans playing God or nature, for example. So over the past few thousands of years, humans have been able to gradually breed a lot of different plants and animals into forms that bear little resemblance to their wild ancestors. And the purpose of that is to make these animals or these plants more suited for our use and for our palate. Over the past few thousand years, humans have actually gradually bred a lot of plants and animals into forms that bear very little resemblance to the wild ancestors. And we can see an example in our dogs. The dogs that we have now are so different from the ancestors of dog wolves. And now we seem to be breeding dogs based on what we want their physical traits to be, as well as their personalities. And so these modern dog breeds that we have now were artificially selected for about 150 years ago. So for example, right, we were able to make a Labradoodle mix by selectively taking a poodle and mating it with a Labrador retriever. And it's interesting because in just these few thousands of years, we've been able to select for dogs into such different types. And so if artificial selection can produce changes this quickly, think of just how much natural selection could have done over the millions of years that it's had to act. Another example is in the plants that we've been able to artificially select. Our modern corn looks absolutely nothing like its original ancestor. And so we obviously have selected based on what us humans prefer in taste, in economic value. But it's really incredible because we've been able to take this plant and change its entire shape, taste, and nutritional value in order to meet what we prefer. So, second question, what fundamental thing has changed between the ancestral and modern corn? So what has allowed it to get to what it looks like now? And the answer is DNA, because we've literally changed the DNA of the corn in order to morph it into the corn that we would like to eat, which is our modern corn. So we come to this interesting idea of, is evolution still going on, right? Because in the past, if an animal does not adapt well to the environment, they get sick, for example. What happens is they would just die because of natural selection and because they are just not well adapted to the environment. But now, because of our technological and medical advances, if someone is sick, we obviously try to treat them. And so it doesn't seem like humans are going through the effects of natural selection. But it's interesting to see that that's not true. A lot of things are still going through natural selection and they are still evolving. So in a lot of places on Earth, species have changed in time spans as short as a few decades in response to human-induced environmental changes. So what we have here is antibacterial resistance, which is where a population of susceptible bacteria that can be killed by the antibiotics we use to fight it has now evolved so that they are mainly resistant to the antibiotics that we've used. And so how that works is when you have a population of susceptible bacteria, it is very rare but possible that there are little bits of resistant bacteria inside there. And so these resistant bacteria could have occurred due to a mutation, but because they are resistant, they don't get killed when we use antibiotics. And so they keep on living, and as they are able to multiply, our antibiotics are not able to catch up to the bacteria quickly. So we don't realize that they are multiplying and they are not resistant to our type of antibiotics. And so as they keep multiplying and multiplying, they become the main type of bacteria, and we are now not able to combat it with antibiotics. And viruses can evolve even faster, which is one of the reasons why it's been so difficult to fight viral diseases like influenza and AIDS. And that's why every season we have to get new vaccines, because we need to be constantly updating our vaccine strain, because we know that viruses can evolve so quickly. Okay, so last lecture we did talk about life on Earth, and specifically what life on Earth is made of, right? So we know that the most common elements are carbon, hydrogen, oxygen, and nitrogen. And we know that life is carbon-based. So Earth is made from more than 20 different chemical elements, but these 4 elements actually make up 96% of the mass of typical living cells. So we know that carbon is a particularly versatile chemical element. It can link together in endless different varieties of carbon skeletons, and it has different sizes and branching patterns and different chains. And we also know that this is due to its valence shell of 4 electrons, which allows it to bond to as many as 4 atoms at a time. And so here we have CH4. Question 3, why is life carbon-based? And we know that it's because carbon is incredibly versatile. We also talked about the different molecular components of cells, and that we have carbohydrates, lipids, proteins, and nucleic acids. So we're going to be talking a little bit more about proteins. And we know that nucleic acids, like we talked about last time, is the basis for storage of hereditary informations. And we can see that the way it's linked is due to the bonding and the relationship between these 4 bases: adenine, thymine, guanine, and cytosine. So a bit more about proteins right now. Proteins are created by the same ingredients that make up DNA, A, T, G, C, right? And so here we have T, C, A, G, and different combinations of the 4 nucleotides are able to create different proteins. So for example, if we have ATT, that makes isoleucine, and if you have ACA, that makes threonine. So we're going to talk a bit more about proteins, but first, let's understand how DNA replication works.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously.
All right. So we're actually not going to finish it. All I want you guys to take from that video, you don't have to know the steps, or the names, but I want you to realize, just how tedious everything is. The fact that every single base here, right, is added on according to its partner base. And so there's room for error, and there's room for copying error, there's room for error due to radiation or mutation. And so it's lucky that our cells, they have mechanisms put in place to correct some of the mistakes, but it doesn't always get corrected. And so if a mistake is made and it's not corrected, mutation can occur. This mutation can have a really big impact on the proteins that we just talked about earlier. So for example, we here have AAG and AGG. So we're going to go back to the other page really quick. So we have our AAG here, and that creates the protein lysine. And then we have AGG, which creates the protein arginine. So if we are trying to code for AAG and we want to create lysine, if there is a mutation and it changes this A into a G, right, then we won't make lysine anymore, we would make arginine. And so it's this idea that when we're making a string of amino acids, that can then create a protein, right? A little tiny base mispairing will be able to create a completely different type of protein. And so if that happens in your body, then a different type of protein will be made, and your chemical composition, your protein composition would be off. And so it can have a huge effect on your body and in the bodies of really any living organism. So for example, Elizabeth Taylor, right, famous actor, she's known as Violet Eyes, right? She's known for her beautiful eyes. And one of the contributing factors for her such beautiful eyes is actually because she was born with a mutation. So she was born with a mutation, the FOXC2 gene. It's a transcription factor that is really important when you're developing. So this mutation actually changed the composition of her gene, and so it changed the way her eyelashes developed. This mutation made her eyelashes really thick. It gave her two sets on top of each other. And so in a way, her mutation was beneficial to her. So in the eyes of society, she was seen even more beautiful than she already was. Right, so are mutations good or bad? We've seen examples of bad mutations that can cause death. We also know that there are some mutations that are actually good. It makes someone more suited for their environment, and that's one of the driving forces for evolution. So the answer is it can be either. It just depends on the environment that the organism is in. All right, so lastly, metabolism. So, metabolism is kind of just a blanket term that refers to the chemical reactions that occur in living organisms. And so it's evolved in providing energy or nutrients to the cell. So we know that the cell's primary purpose is to act as little tiny chemical factories. So like any other manufacturing process, this needs two basic things. It needs a source of material. So you need the material in order to build new products. And in the case of living cells, the key raw materials are molecules that provide the cell with carbon or other basic elements of life. And then the second is a source of energy to fuel the metabolic processes that then break down old molecules and manufacture new ones. So each enzyme is specialized to catalyze different chemical reactions needed in cellular manufacturing. And our source of energy comes from ATP, stands for adenosine triphosphate. It's named that way because we have 1, 2, 3 phosphate groups, and then we have our adenosine group here. So how it works is adenosine triphosphate releases energy by giving up this phosphate group. So as it gives up this group, energy from the ATP can fuel cellular manufacturing. And the interesting thing is that this can actually be recycled, in that after the phosphate group is gone, we have a portion here, and we now call it ADP, adenosine diphosphate. So this ADP can then be recycled, and it can use energy to then assemble ATP. And this ATP can then be used again to fuel cellular manufacturing. So here we actually classify living organisms based on how they get their energy. So first we have our photoautotrophs, which means they get their energy from sunlight, as in photo, and they get their carbon from carbon dioxide from their environment. And so this includes plants and some photosynthetic bacteria. And here we have chemoautotrophs, which are bacteria and archaea that live in pretty extreme environments. So they actually get their energy from chemical reactions that involve inorganic molecules. And then they get their carbon from environmental carbon dioxide. And thus they are autotrophs. So third, we have photoheterotrophs, which get energy from sunlight, but they get their carbon from consuming other organisms or the remains of other organisms. So these are some bacteria and archaea. And then lastly, we have animals and some microbes. So humans, we are a part of this. And we are chemoheterotrophs. So we get our energy and our carbon from food. So what are humans? We get our carbon from food, we get our energy from food, and so we don't make any of it ourselves. We get it secondarily. So we are chemoheterotrophs. Extremophiles. Extremophiles make humans seem like complete wusses. I mean, we need oxygen, a comfortable temperature, proper pressure around us, and all of these things that to us seem like just basic necessities to live. But to extremophiles, they don't need any of that. So extremophiles, they consist of organisms that can survive in really harsh conditions. So like really hot volcanic vents under the ocean, in extremely acidic pools, in radiation sites, and even areas where there is no oxygen. And so one of the most well-known extremophiles are the tardigrades. They can survive in basically any environment, and they are hypothesized to be one of the only living organisms that can survive in outer space. And so in one of the coming lectures, we're actually going to do an accompanied lab that looks at tardigrades. And so what we're going to be doing is going out into our environment, picking up samples of tardigrades. They are everywhere. And then we're going to look at them under the microscope and see just how these crazy extreme organisms function. Okay, so this is the end of today's lecture. Thank you so much for sticking with me. And if you guys have any other questions, email me, I'll be here to answer anything. All right, good lecture. I'll see you guys next week. Okay, thank you so much. Bye.